Well, thank you for inviting me to speak. The title is Environmental Dimension of Antibiotic Resistance, but as Garda said, there's a real crossover between studies of what we think classically of the natural and farmed environment and actually the environments that are within us and within animals. So, excuse me, the human microbiome, sort of the microbial ecology of the gut flora has really come out of the sort of environmental microbiology field. All the molecular tools that have been developed for microbial ecology, molecular ecology, are now being applied to microbiomes. So I'm maybe slightly befuddled. I've got up at 4:30 to come from Cornwall this morning. So I work for the University of Exeter Medical School, which has a base in Truro, the European Centre for Environment and Human Health, and actually near Falmouth on the Penryn campus. There's about 2,500 undergraduates. It's a big centre for evolutionary biology and ecology, and this is the Environment and Sustainability Institute where I have my laboratory. So probably I don't really need to show this, but Garda's just talked about this, but I'm going to blow my own trumpet, which is that 2 of the 7 case studies were research that I've been involved in for the 7 research councils in this cross-council call. The BBSR and the NERC case study was work that I did with Liz Wellington at Warwick and a student, Greg Amos, and then the BBSRC case study is work that I'm doing with Jason Snate with AstraZeneca and some other work that we've been doing as well. So microbial ecology, because it's really, a lot of it is about ecology. So that is the number of bacteria on Earth, which is when we talk about mathematical modelling and stuff like that, is when you've got this, the scales to do with microbial ecology are huge in terms of numbers, in terms of size, in terms of duration, in terms of evolutionary timelines. Maybe 3.5 billion years. Antibiotic resistance, we think, probably evolved billions of years ago. So this is a great quote: "If every bacterium was a penny, the stat would reach for 1 trillion light years." So it's really hard to get your head around these numbers. There's a billion bacteria in 1 gram of soil or 1 gram of feces. And they're all exchanging genetic material, as Jodie's shown very elegantly recently, on a very rapid basis. So yeah, 5,000 species in a gram of soil, and we can only culture less than 1% or 0.1%. So again, we know from molecular phylogenetics that some of the major beta-lactamase clades diverged 200 million years ago. So it tends to be in the media that if we all washed our hands properly, everything would be fine. But of course, it's a much more complex problem than that. We know if you look in 30,000-year-old permafrost, there is DNA encoding vancomycin resistance genes. If you clone that DNA into a modern bacterium, it actually confers resistance. So it's not a new phenomenon, but it's what we're doing in terms of the veterinary and clinical usage of antibiotics, and then how we dispose of those and where the waste goes, that may be having is having the major effect in amplifying resistance. So most of you will be very familiar with horizontal gene transfer, but so there's just this uptake of naked DNA. Some bacteria actually excrete DNA, or when cells die, the DNA can be taken up and integrated into the genomes of other bacteria. There's the phage transduction, which is where viruses can move DNA between bacteria, and then this sort of pseudo-like sex with conjugation, where you actually get pass or transfer of genetic material between bacteria. So we're just really beginning to realise the scale on which this occurs, and actually the range of bacteria, or the evolutionary distance between bacteria that can actually exchange. So one of the really crucial things is under this selection of veterinary clinical antibiotics and the residues that are present in the environment, how do these new genes get into human pathogens? So, we see all these extended spectrum beta-lactamases and carbapennamases. Where do they come from? But they come from the environmental metagenome. But where does that transfer event happen? Does it happen in a polluted environment? Does it happen in the human gut? Does it happen in the animal gut? Well, we don't really know. So this is a great paper by Uli Klumper from Bart Schmet's group in Copenhagen. You probably can't see very well, but effectively they've used Pseudomonas and E. coli, which they've cloned a red fluorescent protein into, and then they've got these resistance plasmids, which they've actually then cloned a green fluorescent protein onto the plasmid, but the plasmid is under a repressor in the chromosome of the host cell, so it's switched off when it's in the donor that you can mix with a population of bacteria. But when the plasmid is moved or is transferred into another bacterium, the red fluorescent protein is switched on. So using flow cytometry, they can then separate those and do genomics. And what they've shown in this recent paper is that these broad host range plasmids that confer antibiotic resistance that we find in gram-negative pathogens can transfer more or less within the whole of the microbial world. So they've transferred to 11 bacterial phyla, including gram-positives, actinobacteria, whatever. And they also show that the relationship between the phylogenetic relatedness, so how closely related the bacteria, the recipient and the donor are, has no effect on the frequency of mobilisation. So basically, I went to a conference with about 100 people, all working on environmental aspects of resistance. You know, and people are saying we have to accept that genes are being mobilised everywhere all the time. But I mean, that's a bit of an exaggeration, but at least there's the possibility that that is occurring. So, and this obviously has huge implications for classical strain-based epidemiology. So, for example, looking at links between agriculture and clinical environment, it's all very strain-based. One of Jodie's papers, which I think you talked about at the SGM, with the gene transfer, I'll talk about it a little bit in a second. You know, this new data that we see on mobilisation of genes, it sort of turns the whole sort of idea of the sort of limited transfer between agriculture and the clinical environment on its head, really, because we've always been looking at the host strains. We're not really focusing on those mobile genetic elements. So, you know, when we think from a clinical perspective about antibiotic resistance, we think about antimicrobial resistance in pathogenic bacteria that cause infection, which is just this bit here. But of course, most of the bacteria within us are commensals, you know, that are actually beneficial or are not really doing us any harm at all. And then, of course, you know, we don't live in a vacuum. We're connected through food to food animals, to slurry and sludge through sort of crops and application to crops and then back to humans, and through water. So, and also we know that there are residues of veterinary and clinical antibiotics in all these environments. So in soil, because of addition of slurry and sludge, which contain antibiotic residues, we can have milligram per kilogram concentration, so it's microgram per gram. So it's quite high, really. And in water, we've got nanogram per litre to microgram per litre. So it's quite low, but then some recent research is suggesting that you can have selection for resistance for fluoroquinolones, particularly down to so what is it, picograms per mil or nanograms per litre. So these concentrations of antibiotics that we now see that can select are similar to the concentrations that we see in the environment. So one of the key questions is, do antibiotic residues in the environment select for resistance? At the moment, the evidence is that it is probably happening, but actually measuring that is or quantifying it is quite a difficult thing. So in the paper, I think the other day was this article about livestock-acquired MRSA in our food, the CC398. This I just wanted to mention. This is I could only find this pre-production publication version of Jodie's paper, but effectively looking at that CC398, looking at isolates that are human-adapted and pig-adapted, when those strains are put into a pig, then the human-adapted strains acquire mobile genetic elements from the pig-adapted strains within 4 hours, including, is it phage-mediated and plasmid-mediated. So, you know, it's always been that, oh, well, it's not a risk because, you know, the strains in animals are animal-adapted and, you know, they have to be human-adapted to be a problem. But now, of course, within hours, it's been demonstrated that they can acquire the genetic machinery to adapt to a different host. So I think as time goes on, we're going to realise this is much more of a complex subject than we ever imagined. And just to show that you don't actually have to look very hard for resistance in the food chain, this is a letter from actually an honours project student in Liz Wellington's lab when I was working in Warwick. So we just took a few chickens, swabbed them, isolated on third generation cephalosporin. Of course, they were covered in third generation cephalosporin resistant bacteria, but carrying, and the percentage of those were carrying CTXMs, but they were in this Rarnella aquatilis, which is like a sort of more environmental Enterobacteriaceae species. But if you were screening these food samples, what would you look for? You might look for E. coli, you might look for Salmonella. So it's this sort of hidden resistome. So, you know, it's all these commensal bacteria carrying genes. So this was CTXM3, which is not the dominant CTXM in the UK, but it causes, it confers resistance to epidemic infectious E. coli strains in other parts of the world. So, you know, every time you eat something, you're connected to the environmental and agricultural metagenome or resistome, however you want to say it. So this is a conceptual model of the sort of the whole problem from a sort of a public health perspective. This is called this DIPSY model. It's developed by George Morris from Glasgow. So you can think about the drivers, that's the use of antibiotics and biocides in the clinic and in agriculture. Then we've got introduction of these resistant bacteria from humans and the pharmaceuticals and personal care products into the natural environment. This is from an environmental perspective. The state is actually what's going on in that natural environment. You know, what are the interactions between bacteria? Is there selection for resistance? Do they just die off and we just have there's a window for exposure? But exposure here, so food chain, contaminated drinking water, direct exposure through maybe through recreational use of coastal waters, which is something I'm interested in. Then the infect, which is colonization or infection, and then you've got this action. So you've got these critical control points where you can act on this cycle. So some of the work that we've been doing looks at different areas of this, and these are the three areas of research that I focus on. So dissemination of resistant bacteria, mobile genetic elements, resistance genes in the environments. So you know, on a sort of a geospatial scale, so on a river catchment scale, but also in between individual bacteria. In situ selection, so evolution, and then exposure. And there's actually much less work done on this human exposure to environmental reservoirs, partly because it's a little bit difficult to get a handle on, but I think that there are ways that we can approach it. So dissemination, this is from Greg Amos's work. So this graph just shows downstream and upstream of a wastewater treatment plant for E. coli and for enterics in 2009 and 2011. So basically it shows that downstream of wastewater treatment plant, there are much higher numbers, and this is of third generation cephalosporin resistant for resistance, much higher levels downstream in E. coli and in enterics than upstream. And this basically just shows, this is the CTXM15 gene, so that they're actually, a lot of these bacteria are carrying CTXM15, which is the most important extended spectrum beta-lactamase. These are different genetic contexts, and they're all indicative of a mobilization event. So Greg found that there were, I think, something like 9, or I can't remember exactly, but 7 or 8 or 9 new genetic contexts that he described from this one site downstream of one wastewater treatment plant that have been reported from all clinical isolates in all the world in terms of genetic context of CTXM15. So either we're looking harder than anyone's looked before, or there is recombination going on inside wastewater treatment plants or in polluted sediments. So, you know, this is this sort of circumstantial evidence. So now we have to drill down and actually look at experimental work to try and document exactly what's going on at environmental concentrations of antibiotics. So this is another graph which shows the same upstream and downstream from a wastewater treatment plant. So these are the downstream samples, these are the upstream samples. This is molecular prevalence, so in all of those billion bacteria per gram, so one doesn't sound very many, but that's 1% of a billion per gram, are carrying these class 1 integrons, which are mobile genetic elements which can integrate not only biocide resistance genes, but a whole range of antibiotic resistance genes. So this is like 300, 600, 900 meters downstream and the same upstream. And what we see is there's about an order of magnitude difference in prevalence downstream than upstream. And these integrons are involved in clinical resistance in gram-negative pathogens in the clinic. So this is we did with CEH This is still work with when I was at Warwick, with Liz Wellington, and since looking in the Thames catchment. So we looked at about 12 or 13 sites at 4 times in the year. This is the Integron prevalence. It varies dramatically by about 2 to 3 orders of magnitude. And when we look at each site and we look at the distance from each wastewater treatment plant, the size of the wastewater treatment plant, so the number of people that it serves, and the type, whether it's tertiary or secondary treatment, we use that in a statistical modelling exercise. Basically, we could predict about 50% of the variance based on proximity, type and size of wastewater treatment plant when we introduced the land cover on a 2 kilometre sort of corridor either side of the river stretch. Then we can explain about 80% of the variance in Integron prevalence in sediment just with those quite simple geospatial analysis. So this is from a paper by Tong Zhang, who works at Hong Kong University. These are different sewage sludge samples from the sewage treatment works that serves Hong Kong, which serves about 5 million people, very cosmopolitan. And these are all the different types of resistance. So this is tetracycline, sulfonamide, polymyxin, multidrug efflux, chloramphenicol, beta-lactam, aminoglycoside, whatever. So, but you get the message. So if you look in sewage sludge, you find huge range of resistance genes to all the known classes. So we've just recently received funding to extend that Thames catchment model using the single marker gene to using next-gen sequencing to look at a much larger subsample of the population using shotgun metagenomics. So we can look at all 3,000 known clinical resistance genes. We're also going to look at inorganic and organic water chemistry and also look at population structure. So again with AstraZeneca, with Jason Snape, we've got one PhD student and another one starting in October to use sort of experimental evolutionary techniques to look at selection. So we can use fluorescently marked isogenic pairs. So they're identical apart from one has a resistance gene and the other doesn't. So you can then compete those against each other at different concentrations of antibiotics, and you can work out what the minimum selective concentration of the antibiotic is. And then you can ask, well, is that concentration greater or lower than the concentrations that we see in polluted environments? So we're doing that in sort of simple single species systems, and then with the new PhD we're going to move to more complex systems looking in sediment communities. And just recently we've had one of these innovation grants funded, which is to use anaerobic, to look in anaerobic communities at evolution of resistance. So these are the, sort of the simplest gut model that you can get really. And interestingly, we're hoping to recruit this Uli Klumper from Copenhagen, who's done this work using these engineered strains, looking at gene transfer, and apply that to these anaerobic fermenter systems. So we can actually look at what genes are transferred from a pathogen to the metagenome and what genes are transferred from the metagenome back to a model pathogen. So, and you can look at that under different selective regimes. So exposure, just to finish off, so we've recently had a paper in Environment International which looked at exposure to third-generation cephalosporin-resistant E. coli in bathing water around the UK. So we took 100 samples from the Environment Agency's bathing water sampling programme. We quantified 3GC-resistant E. coli, and Anne Leonard, who did this work, downloaded all the data from all the 500 bathing waters around England and Wales for the 20 weeks of the bathing season, and then did a very simple calculation using the mean colony forming unit of E. coli per 100 mils, and she did a sort of a quasi-systematic review to look at the ingestion rates for different types of recreational water use, and we come up with about 6 million recreational sessions occurred that resulted in the ingestion of 3GC resistant E. coli. So if you actually increase that to all bacteria for all resistant types, it goes up from, this is about 0.1% of E. coli are 3GC resistant, but actually about a third or a quarter of all E. coli are antibiotic resistant to one or more antibiotics. So the potential exposure is massive, but what do we know the outcome of this exposure? Well, no, we don't. We don't know what exposure to 1 or 10 or 100 bacteria means in terms of colonisation, let alone infection. But obviously colonisation is a risk factor for infection. So that's one area that I would like to look at in more detail. So going back to this sort of conceptual model of the problem, so we can look at reducing antibiotic usage, improving waste management. We need to increase the evidence base on the sort of the state of this environmental reservoir. Is the evolution of resistance being driven by anthropogenic pollutants? And we need to generate more data on exposure and the risk of colonisation and infection, which is the effect. And unusually, for when you look at all of these conceptual models that can be applied to smoking or to obesity or to all of these things, antibiotic resistance is unique in that the effect is infection. So what do? You use more antibiotics, which provides this feedback loop to the drivers. So it's the same, the thing you're doing to treat, as we all know this, but actually when you see this sort of circle here, I think it's quite interesting to, it makes you think really. So we've done quite a lot of work in here, and increasingly we're doing some more here, but this bit here, I think still there's a lot to understand there. And of course we need to parameterize and predict the impacts of our mitigation strategies that we hope to implement, because particularly in wastewater, I think it's been quoted as something like £30 billion to upgrade all secondary sewage works to tertiary, and the amount of power that would use would be huge. So, we need to sort of do this economic cost-benefit analysis as well in order to get action on some of these mitigation strategies. So that's it. This is the group at the moment working on antimicrobial resistance. These are the people that did other bits of the work, and I'd like to thank BBSLC, AstraZeneca, and now, just very recently, MRC and NERC for funding. Thanks very much.